Hello everyone, hope you all are doing well. So today we are going to discuss the next question of our agriculture optional answer writing practice series. And the question for today is, what do you understand by spontaneous mutation, induced mutation and mutation rate? Okay, and describe the practical application and significance of mutation in cereal crops. So this is the question, and let's see how we can approach the answer of this question. And I think mutation is one of the most important topics, almost asked every alternate year by UPSC. So please prepare it well. So you can simply give some definition of mutation. So what do you understand by mutation? So mutation is the sudden heritable change in the phenotype of an individual. In the phenotype of an individual. So it is the most simplest definition of mutation which can be written. So you can introduce by simply writing it. Okay, and now you can simply move to the body. So we have to discuss three words, that is spontaneous mutation, induced mutation and mutation rate. So first of all we will discuss spontaneous mutation. Spontaneous mutation. So what is spontaneous mutations? So these are the natural changes in DNA sequence that occur without any external influence. Natural changes in the DNA sequence in DNA sequence that occur without any external influence, without any external influence, that is without any artificial or any influence. So you can give examples here, not examples. How does it occur? So it occurs due to errors during DNA replication, due to errors during DNA replication. It can be due to the natural changes or natural chemical changes in DNA. Natural chemical changes in DNA. For example, you have heard about deamination. Okay, deamination. So it is due to the changes in protein structure of DNA. Various time transposable elements or mobile genetic elements leads to its change. Various time it is due to transposable, or which can be transported, or mobile, that is moving from here and there, which has ability to move, ability of movement, that is mobile genetic elements. So these are the things which can lead to spontaneous mutation. And one thing you should remember, that spontaneous mutation occur at a very low frequency and are random in nature. They occur in very low frequency and are random in nature. So this you can mention in spontaneous mutation. So this was about spontaneous mutation. Now we will see the second term, that is induced mutation. Induced mutation. So what is this induced mutation? So these are artificially generated by exposing organism to. Induced mutations are artificially generated by exposing organisms to physical or chemical mutagens, physical or chemical mutagens to increase mutation frequency. MF is mutation frequency. So what is it is done artificially, or it is generated artificially by exposing organisms to physical or chemical mutagens to increase mutational frequency. So what physical mutagens can be used? For example, X-rays. All the rays will come under physical mutation. X-rays, gamma rays are there. We have UV rays are there. So all these will come under physical mutagens. When it comes to chemical mutagens, there are certain chemicals such as EMS. EMS is known as ethyl methanosulfonate. I'm writing the full form also. Ethyl methane sulfonate. So this is EMS. You can sodium azide can be used. Okay, nitrous acids can be used. So these are all induced. These are all chemical mutagens, and this method is commonly used in mutation breeding to create genetic variability. So it is used in mutation breeding to create genetic variability, that is variation in the genes. So this was the second term. The third term is the mutation rate. So what is this mutation rate? So it is the frequency at which a specific mutation appears. Frequency at which a specific mutation occurs, or you can say a specific mutation appears in a given gene or organism over time. In a given gene or organism at a or organism over time. So this can be the mutation rate. So it is kind of at what rate mutation is happening. The simplest definition would be what rate mutation of is happening. That is at what frequency specific mutation are appearing in given gene or organism over time. Okay, so the rate of mutation. So it is generally expressed in two terms. First one is the mutation per gene per generation, and another is the mutation per base pair per cell division. Okay, so these are the two things on which it can be expressed. For examples, you can also mention that the natural mutation rate in plants ranges between, so mutation rate, it is natural mutation rate in plants ranges between 10 to the power minus 5 to 10 to the power minus 8 per gene per generation. Okay, these things. So this is what mutation rate is, that is the frequency of occurrence of mutation. So this here we have completed the first part of the question. Now the second part of the question is describe practical application and significance of mutation in crops. So we will simply describe it, practical applications / significance. So both are similar things only, same points will be in both the things. So you have to adopt as per it. So first one is will be the development of new varieties. These new varieties can be of various qualities. Okay, it can help in high yield, it can help in disease resistance, stress tolerant, it can increase yield, it can lead to disease resistance, stress tolerant, any feature can be there. And you can give example of some cereals here, such as in wheat there is a developed variety of Sarbati Sonora. Sarbati Sonora. It is a mutational variety of wheat. Okay, we can also give example of Jagannath. Jagannath is a mutational variety of rice, paddy, what you want to say, and it was developed by OUAT. Okay, the Odisha Agriculture University. Now, second, it can obviously induce genetic variability. induce genetic variability, that is they can as you see that in most of the cells they are self pollinated, so there is a lot of homozygosity on it. So due to mutation we can broaden the genetic base, which can help in further breeding programs. It can also help in improving quality traits. Okay, improve quality traits. For example, it can increase grain size, it can increase the grain quality. For example, you have there is basmati rice, so various basmati varieties are also developed from mutation to increase the quality of grains. Next, it can be helpful in stress tolerance or stress resistance, stress slash disease and pest resistance. So it can be used in both stress resistance and also in disease and pest resistance. There are various diseases, for example, ruts are there, smuts are there in wheat, there is blast and bacterial blight in rice. All these things can be treated with it. All the things can be resistance can be provided with all the diseases, pest and stress in cereal crops. You can give example of some pests which we can provide resistance upon. You can give example of here stem borer or anything. Okay. Now, it can also help in breaking sterility, or you can say inducing sterility, breaking or inducing sterility and overcoming self-incompatibility. Okay, so it can be helpful there. So these are the some of the practical applications and significance of mutation. I have given you 5-6 points which you can use in your answer. So I hope you understand all the parts of the question and you will be able to write a good answer now. In conclusion, you can write how mutation can help us in improving our breeding programs, how it can offer a precise and rapid crop enhancement solutions. So all these things can be mentioned in your conclusion. So that is all for today. If you like it, please press the like button, please do share and please do subscribe. It takes a lot of hard work to make such videos for you on regular basis, and if you like, if you share, if you subscribe, it motivates us to make more such videos for you regularly. That is all for today. Have a nice day. Thank you.